What is DNA microarray? DNA microarray is a tool used to determine the expression level of gene from an individual. Usually, microarray contains a probe of biochips consists of either glass or silica. These types of surfaces are used as a spot for single-stranded DNA to bind with the complementary base pairs. The binding of DNA with its complementary fragment is called DNA hybridization. It is a process to produce a hybrid molecule of double-stranded DNA from two different sources of single-stranded DNA. Basically, DNA microarray involves two types of cells collected from a particular individual. These healthy and abnormal cells are crucial for determination on which genes are active and inactive so that the origin gene and its function can be justified. Then, a color-coded with fluorescent text used to make complementary DNA, called messenger RNA, is isolated from the cells. Usually, the green tag represents healthy cells and the red tag represents abnormal cells. Once the complementary DNA is completely made, it is then applied on the microarray probe for hybridization with its complementary base pair. After binding, the result will be analyzed and interpreted based on the color form on each spot.
One of the applications of microarray is to analyze gene expression level in the cells. This can be used to characterize and classify diseases based on the expression level of genes tested. In order to measure gene expression level, the RNA from the cell of interest is first labeled with fluorescent. An example of a disease that can be detected using microarray is cancer. It is possible to carry out simultaneous genetic testing, which can be used to detect tumors by comparing different patterns of gene expression levels in cancer patients compared to normal patients. Apart from that, this method can also be used to identify genes associated with cancer in the cell. Gene expression analysis and genotyping can also be used to design new drugs.
These are the examples of analysis using microarray. The example of analysis using microarray is shown in this journal entitled Tumor Classification by Partial Least Squares using Microarray Gene Expression Data. It is an important application of gene expression microarray data which includes classification of samples into categories such as type of tumor. Microarray enables simultaneous monitoring of thousands of genes expressions per sample. It is characterized by many measured variables on only a few observations. There are procedures in order to classify human tumor samples based on microarray gene expression, which would include dimension reduction using partial least squares (PLS) and classification using logistic discrimination (LD) and quadratic discriminant analysis (QDA). Next is dimension reduction technique, which includes partial least squares (PLS), and it is useful as a predictive modeling regression method in the field of chemometrics. This can be achieved by extracting and constructing k components in the predictor space to optimize a defined objective criterion. Human tumor samples involved are listed below. After dimension reduction by PLS, the high dimension of P is reduced to a lower dimension of K gene components. LD is used to classify a sample as tumorous, which denotes by y equal to 1 if the estimated probability of observing a tumor sample given the gene expression profile X is greater than the probability of observing a normal sample with the same gene profile. The next classification method is quadratic discriminant analysis (QDA). It is based on classical multivariate normal model for each class. Below is the example of formula that need to be used in order to obtain quadratic discriminant analysis.
Proceed with EST, express sequence stack. EST is basically a short piece of nucleotide sequence that often represent any part of a given gene, typically within range 100 to 700 base pair. It can be obtained randomly from cDNA clones and used to identify expressed gene. From the figure 1, it shows that the EST range usually between 400 base pair. The most important step in EST is producing cDNA library. First, mRNA will be harvested from sample and converted into cDNA, and then clone the cDNA into vector to obtain expressed gene library. And lastly, the cDNA will be sequenced. mRNA can be obtained from organism, tissue, or cell line. It depends on what type of gene you want to identify and the reason for generating EST.